Okay. A couple things I've noticed is that some of you are already turning in your choice assignments. 20 points for Gryffindor. That is awesome. Keep them coming. Some of you have started racking up points. Any questions about syllabus stuff, class structure, material from last class period before we get started today?
I have a question.
All right.
So I didn't know which book to buy, so I ordered it on the Amazon Prime. And so if I don't have it yet, still, is that okay? Just for a couple more, maybe like 1 or 2 more days.
Yeah, and I've got the reading for this week on the website.
Oh, okay. Perfect.
Yeah. So just check under week 1. There's a link to it.
Cool. Thank you.
Anyone else? Okay. Well, let's get started. I'll share my screen. And remember, I can't see little electronic hand raises or the chat while this slideshow is going, so just unmute and yell at me if you got a question. Okay. So as we move on in the semester, we're going to start talking about mechanisms of evolution. I know you covered some of this in 1610, but we're going to hit it hard, especially looking at phylogenies, looking at how variability, heritability, and selection coefficient essentially lead to the phylogenies we see now. We'll see how evolution is definitely not goal-oriented. Get a refresher for you on binomial nomenclature, that's scientific names, how to name things, different size groups, all that, and then work on how to actually make phylogenies. Okay. But first, I have a cautionary tale for you. So you're going along semester and you're like, this is really easy, it's going to be great. Boom. Well, this is really jankety video here, but what does this represent? You are the skateboarder in this story. The road
is it maybe like failing a test or something?
The car is exam 1. Yeah. So that car is exam 1, and that dude walking over there to say "I told you so" is me. All right. So this is to scare you a little bit. Don't get complacent just because you turned in a few points. Don't think that you don't need to study. Do your readings before class, all that kind of stuff, or you will wind up in this position. It happens pretty often. Students get complacent for that first 3 weeks, and then exam 1 comes and they get a 55 on it, and immediately after exam 1, this is the most contact I get all semester from students, is right after exam 1 saying, what did I do wrong? Okay, I can tell you right now what you're doing wrong is not reading ahead, not doing the questions in the back of the chapters, all of those questions, and not asking questions in class. Pretty much it. You've also got study groups being organized by Emma. Make sure to take advantage of those. Okay.
So what we ended up building was basically a petri dish.
Why is this
except
flickering?
That it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands.
Okay, it's flickering. We're just going to have to deal with it, I think. Hang on a second.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic, just input that. There's barely more than people like to survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. across the top of it, pour some thin agar that bacteria can move around it. The background is black because there's ink in it. It appears white. First, you see they spread in the area with no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic. It spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they cause mutations to make it 10 times as much. Repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Okay, I apologize for the seizure-inducing flickers there. This little video comes from somebody's unmuted and given feedback. Check your mics. It comes from a pretty recent paper in Science, and this is kind of the still image version of it here, where they're looking at antibiotic resistance using E. coli as a model organism, and they built this gigantic Petri plate. Okay? And they put the E. coli in a nice, healthy, happy place, and basically we're tracking, how long it takes for them to acquire resistance. That's not what's interesting about this study. We'll talk about what's really interesting in a second. The fact is that the researchers knew ahead of time that the E. coli would acquire resistance to this antibiotic. This wasn't to see if they would. They wanted to track it in real time. All right, so you can see that the E. coli is growing along. It fills up this first section really fast, and then it hits a wall. That wall is the antibiotic. Okay, but then it does break through. Okay, this little mutant right here, and then we get a couple more mutants breaking in. They turn up, they hit another wall, break through it pretty quickly, hit a third wall, takes them a bit more time to break through, fourth wall, and then the final wall. So this is a ton of antibiotics going from 0, 11 days is what it took. That's not very long. 11 days to go from a little bit of this antibiotic will kill me to I can take 1,000 times what it would normally have taken to kill me.
So I have a question about that. So how, with E. coli, like getting used to antibiotics and stuff like that, because obviously the antibiotics are designed to like not let that happen, but do they provide resistance just for the fact of getting used to it, or like how do they adapt to that?
That's the perfect question. They don't get used to it at all. They evolve resistance. And so a particular E. coli cell at the beginning out here, it can't survive in that. And it's not even going to live long enough to find out if it can, okay? Because E. coli, they're dividing, every 20 minutes, basically. Okay, so that one's going to die off long before its children, its offspring, so to speak, make it over there. So originally, they did not have resistance to the antibiotic, okay? And their offspring did. This is evolution. You just watched a species evolve resistance to antibiotics in 11 days, really fast. Okay? The really cool part about the study isn't that they evolved resistant to antibiotics. They knew that was going to happen. What was nice about this study is that since they knew it was going to happen, they could track it. And E. coli has a relatively small genome, and so it's kind of cheap to sequence the whole genome. And so that's what they've done. In each one of these little dots, they've pulled out a little section. They've frozen down those E. coli so they can revive them later and study them more. And they've sequenced the genome. Okay. So each of these little dots is a fully sequenced genome of the E. coli that's growing there. Okay. What do you think they found going from here in the dark blue to here in the red?
I think they would have found a change in the gene that provides resistance against that antibiotic, or a new gene would have appeared that allowed for resistance, either or.
Yeah, they actually found a number of things like that, but the change in genome was pretty drastic over this time. So here they kind of, yeah, question?
Yeah, just this may be a dumb question, but it just occurred to me, is like the range of possible mutations that a species can acquire finite or infinite? If you follow me. Like, I mean, is there like for E. coli, is there like a limited number of possible mutations that they can just randomly get? Or is it just like a totally like unguessable amount of possible things that they could randomly turn into? Or random traits that they could acquire?
Unguessable. Now, they do have a finite number of bases, nucleotides in their genome. And so in some sense, that kind of limits the number of mutations that can happen. But each one of those can switch from an A to a T to a C to a G randomly.
So it's technically finite, but ultimately, like there's millions of possibilities, probably.
Quadrillions of possibilities. Okay. Yeah, astronomical. And that's with a small genome. You get larger genome things like salamanders have, genomes 300 times the size of humans. It's exponential growth of the number of mutations that can happen.
So for like COVID-19, for example, they've been talking about like how there's a new strand or something coming out. Is that like a completely different thing, or is that just like the original one evolving?
It's the original one evolving. I actually just downloaded a bunch of their genomes, and so we can get to that at the end of class with the COVID thing, because it is pretty interesting.
But viruses aren't technically alive, are they? Or I mean, there's debate.
It depends on who you ask. Yeah, we'll get into that next week for sure, especially about viruses. If you ask a virologist, they tend to say yes, they are alive. They do all the same things that living stuff does, but it kind of depends on how you define life. So it gets into a bit of an existential question there. But viruses do evolve, absolutely. They just depend on other organisms to do their reproduction for them. Not too different from most parasites.
One question I actually have about viruses, and I don't know if this is anywhere near like what we're talking about, but as they're so different, like all of the bacteria and all of us, we replicate within ourselves. Viruses, they require an external host. They'll go and mutate someone else's DNA, hijack it to reproduce. Is there evidence that they're of the same like chain of life? Did they split off from early self-replicating molecules, or is that possibly an example of a second abiogenesis?
No, there's good evidence that they're just non-cellular forms of life, more similar to the self-replicating RNA that was found in early Earth before cells.
Cool. So maybe even a potential precursor to like prokaryotes and eukaryotes.
Yeah, and there's an outstanding theory right now, maybe hypothesis is a better term for it, about the origin of the eukaryotic nucleus being viral. Evidence seems to at least fit in with that. But yeah, we'll get more into that in a little bit. The focus right now is kind of on the mechanism of evolution and just kind of how it happens in real time. So to kind of get a bit back on track before the next slide, we're kind of guesstimating what we would see different between the genome of this blue dot here and this red one there, they're still E. coli, right? 11 days isn't long enough to get a new species, but we don't even know what a species is in bacteria anyway, so it gets confusing. But it was, somebody said we would expect some differences in the genes for fighting off antibiotic, maybe, or resisting it, at least in some way. Well, they did this, and they found, well, a whole bunch of stuff, okay, because they have all these genomes to go through, okay. And here are some genes along the x-axis down here, and the number of independent mutation events, okay, and they're color-coded by kind of what those proteins that the genes code for, what they're kind of known for doing. So folate metabolism, stress response, transcription, translation. And so it's not like it was just a single gene was different, you know, from one point to the next. A whole bunch of changes happened, okay? And like here in FOL-A, this gene responsible for folate metabolism, we've got 51 mutation events happening, okay? They break it down in the supplemental material of this paper. If you're good at bioinformatics, you can go in and see exactly which ones were changed and all that. But we've got a lot of non-synonymous mutations and just a few synonymous. What do I mean by those terms?
Mutations that give the same benefit but by altering different parts of the DNA?
You're on the right track.
Instead, if it changes the codons, like it changes what protein is created.
Okay, so a non-synonymous mutation means that it actually changes the protein. A synonymous mutation means that nothing actually has changed phenotypically. So you've changed a bit of the genome, one base in there, a nucleotide has gone from an A to a T, for example, but it doesn't have any effect on the organism whatsoever. That's because of our old universal genetic code and the fact that it's got some redundancy built in. Okay. So if you remember, the central dogma of biology is that DNA is transcribed into RNA, which is translated into a protein, right? And the RNA pictured here, these 3-letter codes are a codon, and they tell the ribosome to make a certain amino acid, or to add a certain amino acid there to this protein chain that's being built. All right, so UUU gives us this amino acid. So does UUC. Okay. So this would be a synonymous mutation, right? A U changing to a C, you still get the exact same amino acid. And so that's what they're talking about here, is that some of these mutations could not have any effect on the fitness of the bacteria, but some of them could. So they kind of broke those down and looked at only the ones that could have an effect. And that's what they're showing here. It's these mutation events that actually change the phenotype in some way. They changed the protein that was being made by changing at least one of the amino acids in that protein sequence.
So is it then that the non-synonymous mutations are the ones that actually do change the phenotype?
Absolutely. Yeah.
Okay.
In most cases, biology is really complicated, right? And so it's easier in bacteria because they don't have introns and stuff like eukaryotes, big non-coding regions. It's easier to say that, yes, that's a phenotypic change because it does change the shape of the protein in some way.
So were the non-synonymous mutations the one that developed the resistance to the antibiotics then?
Yeah.
Okay.
A synonymous mutation it wouldn't have any effect on antibiotic resistance whatsoever. And these mutations are happening all the time. Okay.
And
so
what causes mutations again?
Copying errors. Yep. So, you go through mitosis right before you divide as a bacterial cell, and you're making hopefully a perfect copy of your genome, but it's never perfect. That's a mutation. Just an accidental copying error during division.
Do you sometimes need...
Sorry, go ahead.
Oh, sorry. Do you sometimes need synonymous mutations in order to get a non-synonymous mutation? As in, like, 3 of those, and then finally one mutation sets it off and changes the phenotype?
Yeah, so a synonymous mutation can kind of set up for the next mutation to become non-synonymous. Absolutely. But by itself, it doesn't have any phenotypic change.
Are there genetics involved in how many synonymous mutations a person has or a species has? Like, do certain species have more than others?
So different species have different mutation rates. Absolutely. Bacteria are better at DNA proofreading than people are, for example. And so every time we do mitosis, we have a greater chance of having a mutation than bacteria does. And that's just because of the DNA proofreading proteins. Our version is not as good.
Were nonsense mutations tracked in that study as well?
Yes, nonsense mutations were, but those just truncate proteins and stuff. And so they didn't find... Typically, a nonsense mutation just kills the organism.
Okay.
And so that's probably what was going on there. They didn't find them in the ones that made it to the end.
So in the antibiotic study that we were looking at, so in order for all, like, did there have to be a E. coli that was already adapted to the resistance for them, like, not all to die? And then those ones just, like, multiplied and then they ended up all being resistant, or how does that work?
I love that question. Let's circle back to it.
Okay.
Because you're onto something.
Okay.
Okay. Are we clear on the genetic code? So this is just like a refresher from 1610. All right. So let's circle back to it here. And I think this gets at your question. I want to ask, where are mutations happening in this picture?
I want to say all throughout, or never
Everywhere.
stopping.
At the branching points?
Every time there's a color change.
Ooh. All right, we got 3 options there. I am picking door number 1, all throughout. Mutations are always happening. You see this picture and you would kind of think it's like, oh, the color change or the branch points or whatever. Those are actually just spots where they were tracking little populations and they took a sample. Okay. Mutations are happening every time, or there's a chance of mutation happening every time DNA is copied. And so from one bacterial cell to copy itself, it's got to copy its DNA first, right? It's got that circular chromosome. It copies that, mitosis, and then cell separation, okay? The mutations happen during that copying process. And so we started out with some bacteria here on the right. We wound up with trillions and trillions of them. That's because of multiplication, right? DNA, the cells dividing mitotically. Every time that happens, there's a chance that one letter in the DNA code, or 2 or 3, gets changed, just accidentally, okay? It's totally random. Okay? We can't predict when it's going to happen. We can't predict where it's going to happen in the genome. It's absolutely random. So mutations are happening all the time in this giant population of E. coli. What about selection events? Where are those happening here?
That happens between the borders of the amount of antibiotic put in.
Yes. Okay, so we've got the line here. This is a selection event. All right, so this is really important for understanding evolution, is that random mutants are out there all over the place. Every population is not genetic clones of each other, right? Every member of that population, there's like tons of different variation, and then there's nature imposing some selection on them. In this case, we kind of faked nature and set it up so we could easily see it, but we made a selection event right here, and that was the antibiotic. And so, no, they didn't have resistance to the antibiotic to begin with, probably, because they hit that wall and they stopped. And only one of them made it through at first. Only this one little group kind of popped through. So what's that say about selection and mutation?
That it's random.
Well, the selection part isn't. Does that make sense?
Yeah.
Okay. So the mutation is random. One of these offspring over here just happened to have a mutation, accidental, that made it slightly resistant to the antibiotic. Now, in its previous environment over here where there was no antibiotic, that mutation wasn't beneficial. In fact, it may come with some cost. Okay. But suddenly the environment changes, and now that's the mutant that gets to live. Okay, so the environment looked at this random variation and non-randomly selected one of those. It didn't do it willfully or anything. It's just an absolute requirement. Okay. If you have variation in a population and an environmental pressure that selects whether some of those get to live or die, the ones that get to live are the ones that get to make more babies. And so the population eventually, in here, all of them are going to have that mutation because they're all the offspring of things that made it in. Okay. Are we clear on this so far?
What are like other examples of selection events that are like pretty common?
They're happening all over the place. I mean, the coronavirus, for example, right? We see every population on Earth is always undergoing some sort of selective pressure. We'll see more examples, like human examples and stuff, next week. Any other questions?
Yeah, so the only way that first E. coli strand made it through was just because it had a random mutation that made it slightly more resistant to the antibiotic, but it wasn't like planned or anything.
Exactly. It accidentally was resistant to an antibiotic that it had never seen before.
Okay, gotcha.
And so it was allowed to pass through that barrier. And since it was the only one that was allowed to pass through, it gets to make copies of itself. And those copies are very, very likely to have that same mutant because E. coli actually is pretty good at copying DNA. It's pretty rare that it has copying errors, mutations. But it multiplies a lot, and so it rolls the dice a bunch.
And with that top and bottom out of the three on the right, the top and bottom just stop eventually. And I'm assuming that's because all of their descendants died before any selective mutations could happen.
Yeah, absolutely. And so they only made it into one, this far here on the bottom. And just got outcompeted by other groups that had better mutations, for example. Because remember, it's not just one mutation that they had that got them in there. It's this whole suite of random dice rolling constantly every time that they duplicate. And I can't emphasize this enough. It's totally random, but the selection is not random. Okay.
So those like small or like random heritable mutations, is that why people are affected differently like when they get COVID?
Yeah. Yep. Same for all kinds of human diseases. Of course, there's other components in there too, like your body mass index, your age, that kind of stuff. But it's definitely a genetic component to that. Because some people get COVID at 25 years old and instantly have strokes and die, right? So it's acting on our bodies differently. And this brings us to a scary thought. The post-antibiotic nightmarescape that awaits us all very soon. I'd recommend, if you're interested in this, doing the Your Choice assignment, the podcast about antibiotic resistance that kind of goes into this. Sam Harris speaks with a doctor whose entire job is essentially dealing out antibiotics to other doctors and kind of signing off on what patients can get them. I mean, you see the problem. This is 11 days, and E. coli is now completely resistant to like tons of this antibiotic in just 11 days. What's that mean for this antibiotic? It doesn't work anymore. All the E. coli that's left in this particular petri dish is totally fine with that antibiotic. And so if you had this infection, you got a problem. All right. And that's happening all across the world with antibiotics. So let's take a look at the most famous antibiotic. The first one that we actively produced as humans.
Isn't this why the doctor tells you to take all of your antibiotic and not flush it down the toilet? Because it leads to problems like this.
Well, think about it. If you're taking an antibiotic, you're setting up an environment in your body to select for the ones that can survive it.
I have a question about the antibiotics. So theoretically, how much does your own body fight off the
like a bacterial infection?
So like,
I mean, it's
even
gonna
though that they broke through all those levels of antibiotics, does that mean that we could technically still be on like 300 and be totally fine, even though they're breaking through the 3,000?
Probably. I mean, your white blood cells, if you're healthy and have a good immune system, they're in there doing their best, right? But that's another selection event, and bacteria can become immune to attacking white blood cells also.
Okay.
Right. They develop slime coats and stuff to protect them from that. Right. So yeah, it's a problem. Antibiotics are still our best last resort against a lot of infections. It's the reason we have a life expectancy like we do now, right? You go back to the 1800s, you're lucky if you make it to 50. You'd be out working in the farm, get a scratch on your arm, and your arm has to be cut off. That's how we used to deal with bacterial infections. Then penicillin comes along. Okay, so you probably all know the story of Fleming and, accidentally finding that mold on his cultures was keeping bacteria at bay. Well, we started mass producing this stuff because it does a good job. So penicillin, it keeps bacteria from forming a cell wall, essentially. All right, so let me show you how this works. You've got a penicillin binding protein, this PBP, which is kind of perversely named because it's in bacteria, and its job isn't to bind to penicillin. Its job is to go along and make cross-links in the layers of bacterial cell wall. So this peptidoglycan layer. That's what's keeping the cell together, keeping it strong, helping it be a cell, okay? And so it's called penicillin binding protein because that's what we know it from, but its real job is peptidoglycan building. So you can think of PBP that way. It's a protein in bacteria that builds the cell wall. And it's got this little active site on it that binds to these amino acid chains, and when it touches that amino acid, it changes its conformation and built a little cross-linker, right? It's just chemistry and physics at that point, okay? It's all about the shape of this protein and the little active sites. So the bacteria is churning out this protein, and it needs it to make a cell wall. Well, penicillin, the active part of penicillin is this right here, this beta-lactam ring. What it does is it actually has an affinity for the same active site. Excuse me. that the bacteria is using to build the crosslinks, it fills it up. Now what happens?
Bacteria can't bind and form crosslinks, so it can't form the cell wall.
The protein can't do its real job anymore, and so the cell wall never gets those crosslinks, and it just kind of sloughs off. It's like the bacteria gets the mange real bad and it just dies. Okay. So, how does a bacterium, population of bacteria, evolve resistance to this Beta-Lactam antibiotic?
It could shift its receptor site so that the lactam the lactam can't bind to it, or it could possibly even create a new active site.
Yeah. Okay. The easiest thing to do would be just change your active site a little bit so it can still do its job, but it doesn't fit the beta-lactam ring anymore. You don't want to change it too much because then it can't do its job of building the peptidoglycan layers. Okay. But if you change the shape of that active site just enough to where it can still do its job, but the beta-lactam doesn't fill it up, boom, you are resistant. How many mutations do you think it would take? It's a big protein. Thousands of nucleotides go into building that thing.
I want to say 1 if it's the right one, but probably more.
Well, a minimum of 1. Let's just put it that way. It can do it in a number of ways. So here's a few different ways that it's been noticed. Okay, this is obviously heavily studied. Okay, so we get a change. This is the original amino acid sequence up here at the top, and then here are different strains of bacteria that have been noted to be immune to this penicillin. We went from STMK to SAMK. 1 base pair. That's all it took. So how long would that take to evolve?
1 generation?
If it's the right guide roll, 1 generation. Okay, the chances of it evolving that are, 1 to the 10 billionth, astronomical, right? It'd have to be the perfect accidental change in that genetic code, okay? But if you're rolling the dice 100 billion times, that will happen. It's inevitable, okay? And so these astronomical numbers come down to inevitabilities when you're talking about huge populations and bacteria. Okay? So yeah, 1 base pair was enough in this particular strain right there. You change a T to an A. Done.
So with that being the case for antibiotic resistance, is that similar to what we have kind of with COVID and whatever it jumped from, or like chronic wasting disease in deer? Is it still just a few base pairs usually, or is that something that needs to process?
I don't know that I can answer that without kind of bullshitting you, because I don't know enough about the original coronavirus in bats. I promise you, there is a paper that tells you exactly that answer. I don't know. I do know enough to say that at a minimum, it was 1 mutation, and it could have been as few as 1 mutation.
Cool.
This is troubling to us, since we don't want to end up like this person. You've probably heard of MRSA. Any of you who work in a hospital or have been to a hospital have probably heard of MRSA, methicillin-resistant Staph Aureus. So these are basically, Staph Aureus is a bacterium that lives on our skin all the time. And these new strains that have been popping up over the last 50 years or so are resistant to nearly every antibiotic we try to throw at it. Okay. And so we're back to the 1800s. We're doing surgery to remove bacterial infections, hoping it doesn't get into our bloodstream. Staph aureus has been known about for a long time. I mean, obviously, it's one of the most important components of our skin microbiome, described in the 1880s, and first noticed the MRSA variant in 1961. Now we use MRSA to describe every Staph aureus strain that's resistant to any of these beta-lactam antibiotics, these penicillin-like things. So all the antibiotics you've heard of that end with like cillin, amoxicillin, methicillin, penicillin, these are beta-lactam antibiotics, and MRSA is resistant to all of them. Okay, it's got itself a nice peptidoglycan building protein that just doesn't have time for these antibiotics. It doesn't get filled up, but it's still doing its job really well. Okay. Methicillin didn't exist in nature, right? Ampicillin, amoxicillin, these are all made in labs based on penicillin, just trying to get around the fact that things were becoming immune to penicillin. And so we made slightly different versions of the beta-lactam antibiotic, trying to beat evolution, and we're failing, predictably. Okay. At this point, this quote really blew my mind. Penicillin susceptible Staph aureus is a microbiological dinosaur and a source of amusement when identified. I found a paper from 2007, and they got it published in a journal, just noticing that they found a Staph aureus that was actually still killed by penicillin. It was so surprising that they got to publish a paper about that. Almost all the Staph aureus we know on Earth now is resistant to penicillin. Okay. Why?
Is it because we used it so often that, like, I guess everything was forced to mutate to survive it?
Yeah, that's exactly it. Is that we changed the environment and set up a selection coefficient, a selective pressure, basically saying, okay, if you're not resistant to this, you're going to die. And so we use penicillin so much and so irresponsibly that now everything is resistant to penicillin, at least in Staph aureus. Okay. In fact, you very rarely prescribe penicillin anymore because it just doesn't work on most things it used to. We're even noticing this in the soil, these Penicillium fungi that first produced penicillin. It's not working for them in nature either, because all the Staph aureus that's in nature, just in the soil, is also resistant because we've just pumped our environment so full of penicillin for about 50 years.
I have a question.
Yeah.
So let's say we stop using penicillin, if that ever actually happens. Is it possible that in a few years, a very long time, it'll start working again?
Yeah, it's possible. We'd have to stop using penicillin altogether and tell the fungi to stop making it too, which would be tough. But for things like methicillin that no organism produces on their own, absolutely. I think it would be more on the scale of like 50, 60 years before it could start working again. And it would be random as to whether it worked or not, because we'd have to count on bacteria evolving back to a resistant state because of some selective pressure. So we take away methicillin, we'd have to count on bacteria no longer needing to produce it and getting some sort of like fitness benefit from stopping to produce that. Does that make sense? They could just stay resistant forever unless the environment said it's better to not be resistant.
So a theoretical ideal situation is if we found 2 antibiotics that the bacteria had to keep changing from being resistant to one to being resistant to the other, and then once the bacteria got resistant to one, we just swapped to the other, and then back and forth. That would be the ideal hypothetical situation.
Ideal isn't the right word, and I don't know how practical that would be, but if you go to Walgreens and pick up a tube of triple antibiotic ointment, you put it under your scrapes, under your Band-Aid, that's got 3 antibiotics in it at the same time. And that's the idea behind that is, okay,
Okay.
so you think you can evolve resistance to one antibiotic? We'll try 3 at the same time. Much harder, right? Because when you're rolling the dice over and over again, yeah, it's inevitable that you're going to come up with that mutant that is able to tolerate that particular antibiotic. But can it also tolerate the second and third one that's coming at it?
Yeah,
Likely not.
kind of shotgun it.
Yeah, it's kind of a shotgun approach. And so a lot of times with really serious infections, they're using multiple antibiotic courses at the same time for that reason. I mean, that can have side effects on your health, though, if you're taking heavy antibiotic courses.
Could that technically be bad for the future then?
Yeah. Yeah, you're thinking ahead. It's the best we've got right now. You know, modern medicine, of course, is moving to other ways around this, like phage therapy. So genetically engineering viruses that go in and selectively kill bacteria, gene therapy, that kind of stuff. Okay. So let's say we've got an outbreak of MRSA in a hospital in Thailand. It's super nasty strain. It's resistant to last resort antibiotics, things that are literally kept under lock and key and kept for only the most severe cases. We need to know where it came from and what's different about it from these other strains if we have any hope of finding a treatment. And we really don't want to use, you know, a vaccine that, or not a vaccine, an antibiotic that costs $3 billion to produce. We know it's only got a few months of effectiveness before things are immune to it. We want to save that, but we want to know which one of those to use. So what can we do? How do we find out where it came from?
Could you kind of copy that E. coli experiment and place it in a petri dish and see the mutations it makes, and then hypothetically, based on those mutations, look in the environment around wherever in Thailand this was happening?
I think you're like halfway there. Doing the E. coli experiment isn't seeing where it came from, it's seeing where it's going. But looking at mutations, definitely. This is where phylogenies come in, all right? So epidemiologists, for example, this is their approach, okay? So this is the first phylogeny we're seeing here in this class. You're gonna be so good at reading this by the end of the semester, okay? We got a few different parts here. It's basically like a family tree, right? This is the same thing that forensic DNA analysis does to figure out, you know, who the birth father was on Maury Povich or whatever. We're comparing genomes and seeing who was likely the ancestor. Okay, so this is a phylogeny. We've got the ancestral lineage here representing a common ancestor of all these different taxa, these species or whatnot. So there's our common ancestor. We've got a branch point, meaning that this particular taxon took a different evolutionary trajectory than these two taxa. And then we've got what are known as sister taxa. So what this is saying is that the closest relative of taxon B is taxon C. Okay. What's the most recent common ancestor of A, B, and C?
That would be the branch point, wouldn't it?
Yeah, right here. A lot of times when you look at a phylogeny, it helps to think of these as like little pipes, tunnels or whatever. Turn it up on its end, pour water into the two taxa, and see where that water meets. Okay, so taxon A, B, and C, if I poured water down each of these little tubes, the point where it meets is the common ancestor of those three. Okay. We are essentially out of time. And so I've got a short assignment for you. I'm going to pop it up in the chat here, but essentially I want you to make a phylogeny of these candy. So in the chat, I'm giving you a link. Download that. It'll be this PowerPoint slide. I want you to go in and, you know, draw lines branch points. Make yourself a phylogeny like this of the candy, showing which candy is more closely related to the other candy. All right, and we're just gonna have like a slideshow presentation from several of you on Friday when we start up.
Where do we upload that?
It's in our chat link, our chat. There's a little link.
It's just up in the chat as well.
Yeah. I'll throw it up in our main teams also, so you can have it. But it's really just this slide, and you know, these are pictures, so you can move them around, put them close to each other. If you think Special Dark and Milk Chocolate Hershey's are most similar to each other, then...
Do you want us to just screenshot ours and put it in, or how do you want us to upload it?
Yeah, just bring your screenshots to class on Friday, and we can stick them up in the chat and take a look at them. I'm curious as to what you come up with, and where does the banana fit in? Right? Does it go with the banana Laffy Taffy, the unholy abomination, or
Banana's
the mouse?
the disappointing child.
Yeah, it's bad. Like, there's some bad candy on here, so it depends on how you want to group things. Okay. But I want you to start small, find the pairs, and then work out from there. So with these, maybe Butterfinger would show up like this, as it's closely related to the two Hershey's, but in this case I'm saying that these are actually more closely related to each other than either one is to the Butterfinger. All right. There's no wrong answers on this. I'm expecting some of you to make some like theoretical mistakes in how phylogenies work, and that's good. We're going to work through them together to get it figured out.
I'm not seeing anything in the chat. Is it a different chat from the meetings chat in Teams?
It worked for me.
I've had stuff in it before, and I've sent stuff to it, but now it's totally empty.
All right, I'll stick it in our team also. I'll stick it in general, just under a new conversation. All right, so it should be up in there. And that's where we'll pick up with on Friday. Take a look at your phylogenies and then what I think a phylogeny might look like from this. All right, you're all free to go. I'll stick around for a few minutes if anyone has other questions. I've got a meeting at noon, so I got to be out before then. But
I have a question just about MRSA, if that's all right.
Yeah.
Is it that it is resistant to the antibiotic itself, or the antibiotic just can't permeate the phospholipid bilayer or whatever?
It's getting into the cell. It's just not able to find its binding site.
All right.
The binding site has changed. You know what I forgot to mention? I'm going to stick this on the group chat also. There is a faculty candidate giving a teaching demo. I'll put a link to that. And that's, if you write me a paragraph, that's 5 bonus points. Okay, so I'll stick that in the teams. I just ran out of time.
Can you put it in the general one?
Yeah, I'll stick it in the general. And what I'm looking for there is kind of just like pros and cons. I want you to help us pick our next faculty member. You're going to be taking classes from them, right? And so tell us what you liked or didn't like about their teaching.
I have a question. So are smaller organisms able to evolve faster than larger organisms like us, for example?
It has to do with generation time, right? It takes us 20 years to have a kid. It takes E. coli 20 minutes, and so that's why they can evolve faster. not just because they're small.
But typically, do smaller organisms have, or I don't know, just what I've seen is that smaller organisms have shorter generation, or like.
Yeah, I think that's probably fair. Yeah. Okay. All right, I will add that link here in 1 second, if I can freaking find it.
I have a question about the reading.
Yeah.
So, and this class is obviously, super heavy with reading base, and I luckily I enjoy. But how do you recommend us deciding from the massive amount of information in the reading what's important? And
back at the chapter,
because
focus on the chapter summary, and definitely do those questions.
Okay.
Yeah.
Because I like to go make questions beforehand that I can look for throughout the chapter, and I was just so lost with how much information that was. So that helped. Thank you.
Quizzes are based on back of the chapter stuff, for sure.
So I have a question.
And would you recommend? Oh, go ahead.
So, I know we have the PDF of this week's, but the book I ordered from the year suggested isn't coming until January 21st or through the 27th. So do I just need to find another book to order? Or?
27th. Yeah, maybe somebody will let you borrow one or something. The library also has 5 copies, so maybe you can get one. Yeah. All right. So that meeting starts in 3 minutes. I promise I'll try to have a link up on general for you. All right. See you there, hopefully.